All right, welcome back. This is now part 2 of lecture 2 for BIS103. And so in part 1 of our video, we just had discussed how we can use Gibbs-free energy and the equilibrium constants to predict and measure and calculate pathway directionality in metabolism in complex networks of metabolic pathways. We will revisit all of these aspects again as we are moving through our first major pathways over the next lectures. Now I want to switch gears a little bit and give you a little bit of an overview of some of the major chemical reactions that will be important throughout the quarter, and then start diving into our first big metabolite class, our sugars. So let's start with just revisiting a few of the fundamental chemical reactions that occur very frequently in metabolism and that will be important for essentially every pathway that we'll be discussing this quarter and that occurs in biochemistry in general. So I listed 4 reaction types here. These are by no all means not all of them. These are really just the major ones that we will be dealing with this quarter. And so the first one that I have here is sort of the making or breaking of carbon-carbon bonds. And these are called condensation or cleavage reactions. So these are reactions that allow us to break more complex hydrocarbon or higher functionally modified compounds apart at their carbon-carbon bonds. And you will see already later on when we start to talk about the breakdown of sugars, how important these carbon-carbon bonding reactions are. Number 2 here on the list, they also occur very frequently, are internal rearrangements. A really prominent example here are isomerizations. So for example, moving around double bonds between carbon-carbon bonds within a molecule. So the important part here is that these are rearrangement within the molecule. This can be isomerization of double bonds, like I said. It can be moving around hydroxy groups to different positions. And again, these are reactions we'll see commonly throughout the quarter. Number 3 here, group transfers, equally important. These are now across molecules, so group transfers. We can look at the breaking of a bond in a substrate molecule like a hydroxy group or a phosphate group and transferring that functional group over to another compound, then generating a new product. And again, we'll see a lot of these throughout the quarter. And last but not least here, I already touched on oxidation and reduction reactions already, speaking about the catabolic pathways. These are very prominent reactions in the transfer of electrons. And so they're extremely important for especially energy metabolism, but occur really essentially in all of metabolism. Here we go. So I want to touch base a little bit on these oxidation reduction reactions because they're so important and because there's often a lot of misunderstandings in what these actually represent. I think oftentimes it's sort of falsely assumed that oxidation reactions include oxygen and the transfer of oxygen. This is not entirely true. It is true that many of these oxidation reactions do in fact include oxygen, but the real foundational reaction here, what it is illustrated here, and what you have is essentially a transfer of electrons. Okay, so what you can see here in red, we have a reducing agent, also called an electron donor, and this electron donor is an electron-rich metabolite, will give away its electrons, and in this process of giving away, and this is always 2 electrons here, it is oxidized. So oxidation essentially means the loss of these electrons. Okay. This reaction always has to be paired with these electrons being taken up by another molecule, right? So under B here in blue now, you have an oxidizing agent or an electron acceptor, And so this electron acceptor is an electron deficient metabolite. This will take on these two electrons here and is in this process reduced. So keep in mind, oxidation reduction reactions, don't always have to involve oxygen. They do have to involve the transfer of these two electrons, where one metabolite is giving away the electrons, is oxidized, the other one is accepting them, is being reduced. Also keep in mind, right, because of this, these two reactions always have to be coupled with each other. For one metabolite to be oxidized, another one has to be reduced. And we'll see many examples of this. Okay. As a side note here, was typically also this electron transfer would always go hand in hand with the transfer of these hydride ions. There are a few exceptions on how this might work, but it's also important to note. Okay. Just to memorize it, if you want to think about oxidations, right? If you cut an apple and you see the browning, right? This is not meaning that the apple is actually going bad. What this browning means is that they are phenolic type metabolites that very easily oxidize when they get in touch with air. So here, these are really oxidation reactions that do involve oxygen. And so the breakdown and further metabolization of these phenolics here is actually what causing your apple to brown. Just a fun fact. All right. So now that we have touched on sort of some of the really major types of chemical reactions that we want to look at, another thing I want to sort of bring everyone on the same page on is functional groups. These are, again, very important throughout the quarter for us to understand how they relate to what kind of reactions we can use in metabolism and how they relate to energy. Okay? And so what I have illustrated here, this is actually an image from your book, where we have listed a number of different functional groups. So we have the alkane here, so a simple hydrocarbon chain. Once we actually bring oxygen in here, the next one that is further oxidized is the alcohol. If we move along sort of in the chain of oxidation here, the next one is coming to the aldehyde. Or if this is not hydrogen, you have another rest here, it's a ketone. Important to note here, this is where we start to have carbonyl groups right here, right? You see our double bond function. If we further oxidize an aldehyde, we get to our acid. So we're bringing in another oxygen here. We're further oxidizing this carbon to a carboxylic acid. And then our most oxidized form that we have in biochemistry is carbon dioxide. And so what you see here along the lines from this alkane all the way to carbon dioxide is a continued oxidation, right, of these molecules, specifically of these carbons, meaning we are releasing these electrons. That also means, right, if we're releasing these electrons, we're reducing energy. So if you look at this series of functional groups, the one that is highest in energy is actually this one here, the most reduced one, the alkane. And then we are losing energy increasingly through the loss of electrons all the way down to carbon dioxide. Why this is important to look at these functional groups is now how they can actually relate to facilitating some of these reactions I showed you. Okay. So on the right side here, what I'm trying to illustrate is how important these carbonyl groups are, right? So carbonyl groups that are the result of oxidations from the point on of this aldehyde here going further down the line of oxidations. Okay. And if you have such a carbonyl group here, what is actually happening is that the oxygen is drawing electrons away. You have not a loss of electrons, but you have a delocalization. So the electrons that will have been around this carbon here are increasingly drawn toward this oxygen. Okay. So and if this happens, then you can have another effect where now this carbon is drawing, for example, electrons from the next carbon. So all these electrons across the hydrocarbon chain are being delocalized. And as a result of that, the more electrons are drawn to this carbon, you're increasingly starting to weaken these bonds, right? And the result is, for example, that you can have now a double bond form here. Just as an example, this can generate a carbanion. It doesn't only work with oxygen, it can also work with ammonium groups, for example. They're not quite as strong, but it's possible. For the most part, what we will see this quarter are these carbonyl groups here. And one important aspect too that we will see actually in ATP is if you add acids, like this one here, that's an acid, or you add positively charged metal groups here, you can actually increase the draw of electrons from this carbon, and you can increase the relative positive charge of this carbon, weakening the bond even further. And so this drawing away of electrons and the weakening of the bond is what really in the end allows us to do the cleavage of carbon-carbon bonds, for example. So these carbonyl groups are very important to facilitate these reactions. And again, we'll see a number of examples on how this is actually used in metabolism. All right, so this is, I know, very much in a nutshell. I just want to sort of bring everyone up to speed and on the same page for the terminology we're using and some of the foundations. So I highly encourage you, if you feel a bit shaky on these topics of the functional groups, as well as some of these major reactions, to go back to some of your organic chemistry material, but we obviously also can address these in the chat room as well as in the office hours throughout the quarter, so that we can make sure everyone feels strong about understanding sort of the background biochemistry of the reactions we'll be looking at. For now, what I want to do, I want to sort of jump right into our first big metabolite groups, so the carbohydrates or sugars or saccharides. We want to look at how they're broken down for energy in metabolism, but also how we can build them from smaller, simpler molecules and how we can store them away. Okay. So let's start with the sweet stuff. First of all, I want to go through a little bit of terminology of carbohydrates. What are they? Let's start with that. So carbohydrates are defined as either polyhydroxyaldehydes or polyhydroxyketones, right? So if you actually look at these two structures here on the right, number one here, you have your aldehyde here, right? And for number two, you have your ketone function here, right? So you want these two functional groups to be there. And then for the rest of the molecule here, you need a polyhydroxy function, right? So you can ask yourself, what might be the smallest carbohydrate be, right? What you need is you need a carbon with this aldehyde group or the ketone group, and you need polyhydroxy. The smallest way of doing poly would be 2, right? So you need 2 additional carbons to carry those polyhydroxy functions. And as a result, the smallest carbohydrate would have 3 carbons, right? 1 to carry the aldehyde or the ketone group, right here. 1 or to carry the ketone group, and then you need at least 2 that carry the alcohols for polyhydroxy. If you have that present, by definition, this would be a carbohydrate. Again, a little bit more nomenclature. So throughout the quarter, you'll see different ways of how people depict sugars. The one that we will most commonly use is a Fisher projection. And so this is a way of showing carbohydrates that in nature are 3-dimensional, right? To show them in a 2-dimensional way on paper. And so the way the Fisher projection works is that you can see here, right, is sort of just part of a structure. And in order to reflect this now, what is actually happening in the 3-dimensional space is that the bonds here that are facing horizontally reflect these bonds here, these wedges. And so wedges mean that it is facing out of the plane toward you. And the vertical lines here are represented here as dashes. Dashes means they are facing backwards behind the plane of the paper, okay? So if it's horizontal, it's facing forward. If it's vertical, it's facing backward. And I mostly will ask you to just recognize these sugars in the Fisher projection. But it's just one way of representing 3-dimensional chemical structures in 2 dimensions. Next up, the differences between aldoses and ketoses. This, in essence, is fairly straightforward, right? If you have a sugar that has an aldehyde function here, then it's an aldose. If you have a ketone function, such as here, you have a ketose. Also, I would ask you to really understand the numbering system here for our sugars. What you do actually is you look for the functional group, right? In this case here, this would be the aldehyde. In the other one here, it would be the alcohol together with the carbonyl group, right? And so in the aldehyde group, basically you start numbering the carbons closest to the functional group, right? So this would here carbon 1 right next to the functional carbonyl oxygen, and then you just walk down the molecule all the way to 6 here. Right? Same here, next to your functional group, this is your first carbon, and you can calculate down to 6 all the way down. Right? And so we'll see throughout the quarter, we'll be using this numbering. So it would be good to play around with it a little bit, look at some different sugar structures, and try to reconstruct for yourself how you can number these. And again, if you have any questions, obviously the office hours are there to discuss some of these things. All right. Monosaccharides, right? So monosaccharides are carbohydrates that cannot be hydrolyzed into simpler compounds. Okay, these are essentially our smallest sugars, if you will. Right? We look at a few now that can be hydrolyzed in the next slides, but these ones are our smallest ones. And there are a few ones that are listed here. There are many different sugars. These are the ones that I would ask you to be able to recognize. So I'll give you a little bit of a task when we go out there and search for yourself for these structures. And so there are a lot of different tools, one of which I give you here, ChemSpider, you could use, where you simply use a keyword search and you can find that structure. The numbering then for the entire monosaccharide sugars is fairly straightforward. It's essentially just based on the number of carbons. So if you have 3 carbons here, right, like I said, this is the smallest possible carbohydrate, right? You have your aldehyde group here, and then you have 2 further carbons, each of which carry a hydroxy group. So you are fulfilling the requirement for being a polyhydroxy. This is your smallest sugar. If it's 3 carbons, it's a triose. If it's 4 carbons here, it's a tetrose. With 5 carbons, we have a pentose. And then with 6 carbons, we have a hexose. For example, most prominent here is our glucose. Okay. There are higher sugars. We see some of those, but I don't really ask you to know those. All right. About the hydrolyzation, right? So far we had talked about monosaccharides, but sugars can actually also form combinations all the way up to higher polymers, right? And then we call them disaccharides. This would be for 2 sugars, oligosaccharides, larger number of sugars. And then if you have even more sugars combined with each other, you have polysaccharides. And so it's depending on the number of monosaccharide units that you have in there, and also which kind of monosaccharide you have. Right? There's 3 that are really common and that I would ask you to sort of focus on. Maltose, for example, is 1 molecule of glucose and another molecule of glucose combined. And combined here means that we have 1 glucose, the alcohol group, actually building a glycosidic bond with this other hemiacetal here, that carbon. And we'll look into how this works later. This is a glycosidic bond. So sugars can combine with each other through this bondage. Lactose then, we find in milk, right? This is not 2 monosaccharides of glucose anymore. It's actually 1 galactose and 1 glucose. And then sucrose, which is your table sugar, mostly derived from plants. You have 1 molecule of glucose and 1 molecule of fructose. All right. Before we go deeper into the nomenclature, another really important aspect of sugars that has a lot to do with their bioactivity and their importance is the concept of stereoisomers, including enantiomers and diastereomers. And so this means that essentially all these sugars, right? If you think about hexoses, all the different sugars that have 6 carbons have the same molecular formula, they have the same structure, but the atoms have a different orientation in space, right? And this is really important because these differences in the orientation of the atoms in space can be recognized in biological systems, especially enzymes, are often highly specific to only one certain stereoisomer, right? And so this is one way of controlling pathways, that this specificity to the stereoisomers in enzymes can control which substrates are being used by an enzyme. Okay. These different stereoisomers, what they come from is that carbohydrates have chiral centers. What's a chiral center? So it's essentially a carbon with 4 different groups attached to it. So I try to illustrate it here on the right. Right, here's your carbon in gray. You have in yellow here another aldehyde group attached to it. In red, you have a hydroxy group. Then you have in blue an alcohol and a hydrogen. So this is a carbon with 4 different constituents, making it a chiral center. If it would, for example, have another hydrogen here instead of the alcohol, it would not be a chiral center anymore. And generally, the rule is that for every chiral center you have 2 to the power of n stereoisomers that are possible. So in addition to being bioactive, stereoisomers also provide a lot of chemical diversity and potential for different functions. They're very important in nature. Just to give you one example, right? For example, we have here aspartame, right? You probably know this from Diet Coke. It's sort of one of the traditional artificial sweeteners instead of sugars. But if you actually look at the different stereoisomers that we have, if you do have this stereoisomer here, the LL and SS, we don't have to go into this nomenclature, but this stereoisomer here, it actually tastes sweet. If you have a different stereoisomer where essentially this benzene ring here now has a different orientation in the 3-dimensional space, it will actually react differently with taste receptors and it starts to taste bitter. So it's a really good example on how these minor changes in the orientation of atoms in the 3-dimensional space have a huge impact on their bioactivity. A little more on the stereoisomery. There are multiple forms that we can see. There are some forms we call enantiomers. So these are isomeric compounds that are not superimposable mirror images of each other. It's a bit hard to do in a video to show how this actually works. I attached here a little video that illustrates a bit better in 3 dimensions how you can actually understand whether a stereoisomer is a mirror image of each other or not. We compare the enantiomers to diastereomers. So these, again, are stereoisomeric compounds, but they're not mirror images of each other. Why is this important? Again, this difference between whether they're mirror or not has quite an impact on their bioactivity and on the possibilities of the different atoms around the chiral carbon center, on the diversity of possible isomers and the related bioactivity. Then last but not least, a type of stereoisomer that is really common in sugars are epimers. And so these are now diastereomers that differ only at one chiral carbon. Okay. We'll see examples of that. And so as I said, hexoses, other sugars can have multiple chiral carbons, making them really diverse in their structure. For example, for a simple hexose or a 6-carbon sugar, we can have 2 to the power of 4, so 16 different kinds of sugars, all of which could have different bioactivities based on their stereoisomeric properties. And so speaking of the epimers then, right here again, we have a sort of a prime example of glucose. So you have your aldehyde group and then your polyhydroxy side chain. I like to remember glucose just by remembering the hydroxy groups. So it's right, left, right, right. That's sort of a good way to remember it. Now, if you see at galactose, right, we just saw that in lactose as one of the sugars in lactose. It's also a hexose, very similar to glucose. The only way it differs now here is in the spatial orientation of the hydroxy group at carbon 1, 2, 3, 4 right here. So that's the only difference. So galactose is a C4 epimer of glucose. It's also a good way to remember the structure of these things. So you don't have to remember the entire structure of galactose if you learn what glucose looks like and you remember that galactose is a C4 epimer of glucose. You have both structures already covered. Another important aspect here is the concept of hemiacetals and ketols. You probably saw this in the slide just earlier where we talked about disaccharides and polysaccharides and the possibility of sugars to form glycosidic bonds. So the function of hemiacetals and ketols is really critical for the possibility that those bonds can be created. And so the definition here is that hemiacetals are compounds that are the result from the addition of an alcohol group to an aldehyde or a ketone. This can happen in many different kinds of metabolites, but specifically for the sugars, these hemiacetals and hemiketones can also occur within the molecule, so intramolecular, where one of the hydroxy groups of the sugar can actually attack the ketone function or more commonly the aldehyde function of the same molecule. The result is that you form a ring. And this actually happens very commonly and very frequently in nature, that sugars actually occur in their ring form. And this can happen spontaneously. It doesn't even require an enzyme. This is sort of illustrated here. You actually down here, you see the relative ratio of the different forms of glucose that occur in nature. And you can see that while these two different ring forms here that are called alpha and beta are occurring much more commonly, whereas the free form here almost doesn't occur in nature at all. In solution, that is here. And so this ring formation, what is happening here is, right up here, again, you have your aldehyde function. And now down here at the C5, this hydroxy group here, the oxygen has a lot of electrons, right? We have this carbonyl group here. And I told you earlier, right, in these carbonyl groups, the carbon has a partial positive charge because the electrons are drawn away to the oxygen. So now it can actually happen that the oxygen here can attack this carbon and you form a ring. It usually is this hydroxy group here. It is more reactive than the alcohol at the end here. And the upper ones here, it would be very hard to form a ring because there's so much steric hindrance. You just can't, you can physically form the ring. So it's usually the hydroxy group for a hexose here at C5. Okay. And then the alpha and beta versions, they derive from whether this hydroxy group will attack in the front here or from the back of the carbonyl group, right? Keep in mind that while we show this in 2 dimensions, this is a 3-dimensional molecule. So this reaction can happen from the front or from the back, resulting in alpha or beta glucose. One of the advantages here that you can see, right, is if you do this, right, the aldehyde group here is not a chiral center. You have 2 bonds to the oxygen. If you open this up, you actually generate another chiral center. And I told you, right, this is very important for bioactivity. A few more projections here. Right? Mostly what I will ask you to understand and recognize here is the Fisher projection. Given that we do our learning remote this year, there won't be much drawing to be done. So mostly I will ask you to be able to recognize the sugars in their Fisher projection. Another projection now is the Haworth projection. This recognizes the fact that most sugars in nature occur in their ring form. And so what the Haworth projection does is that it essentially puts the Fisher projection on its side. And then every hydroxy group that points to the right here in the Fischer projection points downwards in the Haworth, and everything that projects to the left in Fischer points upwards in Haworth, right? So basically, if you clap this 90 degrees on its side and you form a ring, you can move from the Fischer projection to the Haworth projection. And so these two projections, I would ask you to be able to recognize the sugars in. And then lastly, if you want to actually recognize whether it's an alpha or a beta sugar, right? Okay, when the anomeric carbon, so this is a carbon here where the ring closure occurs, so the former aldehyde group, it's called the anomeric carbon. If the hydroxy group, based on the Fischer projection, is pointing opposite to the C6 here, it is called alpha. And if it's the opposite, so if this hydroxy group would be facing upward, meaning in the Fischer projection it would have pointed here, then it would be the beta option. Okay, so this is how you can differentiate the Fischer and the Haworth projection, as well as the alpha and beta sugars. Last but not least, we also have a Bode-Scher projection here. This again recognizes a more natural state of the molecule and is more precise. But for the purpose of our quarter and our class, I will not ask you to know the Bode-Scher projection. Last concept for today, the reducing and non-reducing sugars. Right? This actually is important for bioactivity, but it has been defined initially arbitrarily through experiments. And so the definition of whether a sugar is called a reducing or a non-reducing sugar is whether or not this sugar actually reacts positively or negatively through a different kinds of tests where people actually use copper or silver in what we call Benedict's, Fehling's, or Tollens reagents. You see an image down here. So basically, if you have a sugar that reacts with copper or silver in these solutions, it will actually give this sort of rusty red color. If it's a non-reducing sugar, it will remain blue. Okay. And so the hemiacetals and hemiketals become really important here again, because in order for a sugar to be reducing, it needs to contain a free or a potentially free aldehyde or ketone function. Why is this important again? So one importance of reducing sugars is that they are more reactive. And so reducing sugars, unlike non-reducing, can form conjugates with proteins, just as an example. And so a really good example is that reducing sugars, so free sugars, actually can form conjugates with hemoglobin. Okay? And this understanding of this conjugation is actually used in some types of blood sugar measurements. So some of you who volunteer or work in hospital may actually have used some of these assays where you can measure the binding of glucose to hemoglobin, and you can use this to extrapolate how much blood sugar a patient has. So again, this understanding of the basic biochemistry of these sugars, in this particular case, the non-reducing versus reducing properties, really can help to utilize this knowledge, for example, in this case, to develop diagnostic tools. How does it work? How can we define this? This is something I would like you to actually pay attention to, how one can deduct whether sugar is reducing or not. The first simplest rule is that all of the monosaccharides, so all of the most smallest sugars here, shown again glucose here in the Fischer projection, are reducing sugars because they do have a free aldehyde or ketone group. Okay? So that's simple. All your monosaccharides are by default reducing sugars. It gets a bit more complicated when you think about disaccharides or higher order polysaccharides. So one example here is our lactose, right? So is this now a reducing sugar, right? Your first immediate answer might be no, because you don't see a free aldehyde or free ketone group, right? But we also said, right, it can be a potentially free group. So the question we have to ask for this molecule here is, does it have a potentially free aldehyde group? So is there a way for this molecule to change that there's an open aldehyde group. And so I'm going to try and actually walk you through this. So let me just switch to the pen. And what I want you to pay attention to now is, again, here the anomeric carbon. So I highlight this just here with a dot. So this is the anomeric carbon, okay. And sort of to complete this, right, this is lacking an H here, this oxygen, and we have to have one more here, right, so that your anomeric carbon has the 4 constituents. What is important to note now is that you have this OH group here, and it's very close to the oxygen within the ring, okay. And so this oxygen is actually quite lonely. It has a lot of electrons. And what can happen with these disaccharides, if they have this constellation, is that this oxygen that is so rich in electrons can steal the hydrogen, all right? Now you're leaving the oxygen here with too many electrons, and what these free electrons are now going to do is they try and find the next best spot to hop to, and that's this bond right here, okay? The result of that happening is that you now have your hydrogen here, right? It just got stolen by this other oxygen. And I told you, right, the electrons here have jumped to this bond. You're creating a double bond. And you still have your hydrogen sitting here. If you look at this now, right, what functional group is this? That now is an aldehyde group. So intrinsically in the molecule, if you have a constellation like this is a potentially free aldehyde group, and therefore lactose actually is a reducing sugar. Another example here are sucrose. Same question, is it a reducing sugar? Right, first answer would be no, we don't see any free aldehyde or ketone groups. But now that we know there are these potentially free groups, can we apply the same system? Okay. What you note now is that you have here at the ring no oxygen function, right? In neither position, neither here nor here, there's an oxygen. Okay. So the same principle of this oxygen stealing the hydrogen away will actually not work, because this bond here, the CH bond, is far too strong. It's too strong for the ring to open. And as a result of that, this actually is not a reducing sugar. We cannot open up this ring to generate a free aldehyde or ketone. So sucrose is a non-reducing sugar. What I want to end on is the last bit is glycosides. This will by no means be on the exam, it's just for a fun fact, but in many cases, glycosides cannot just be combined with each other, but they can also combine with other types of molecules. So in this case here, what we're seeing is a number of sugars that have formed glycosidic bonds to this backbone here. This is actually called a terpene. It's one of the metabolite classes that my research group actually works on. And this molecule here is actually steviol, that you probably know when you went to Starbucks, right? For a while now, we have these natural sweeteners here that you can use. And so this one here is steviol. That's the metabolite that you actually get. There's a variation of them that you get in these natural sweeteners. Okay? And you'll probably also notice the flavor is different from just glucose. Many of you will probably recognize it as much, much sweeter. And some people are really sensitive to a bit of a metallic or bitter aftertaste. And so this aftertaste is actually likely coming from this non-sugar backbone, this terpene. They're typically rather bitter. So the combination probably makes for this slightly different flavor that we're seeing. Okay. But I thought this might be a fun fact for all of you who are using stevia in your tea or coffee. On the last note, because it is so popular, there's actually a lot of biotechnology industry out there. We have used many of the tools I showed you in the first lecture video to engineer, to first understand how stereoside and other steviol compounds are produced in the plant. So people now know the enzymes, they know the intermediates and the entire pathways and how this is formed in a plant. and they can now move these actually into bacteria, and they can produce stereoside in bacteria using metabolic engineering. So it's sort of a nice sort of closing of the circle, how understanding of metabolism and eventually engineering metabolism based on that knowledge can really bring us to new sustainable bioproducts. So that's all for today. Thanks for listening, and I'll see you for the next lecture on glycolysis.